I'm Noélle Germain. I was lucky enough to join Ovid about a year ago, almost a year ago. So this is my first chance to participate in the KIF1A.org meeting, and I wanted to thank you all on behalf of Ovid and me personally for including us in your organization and including us in your community. I can say that over the last year that I've been working with KIF1A, I am incredibly impressed by the tenacity of your community and the amazing individuals that are a part of your group. So today I'm gonna be giving you an update on the work that is ongoing with our incredible collaboration with Wendy Chung's group at Columbia University. Next slide. Okay, so this is just some disclaimers. We can skip to the next slide. Thank you. And so I want to give you all, for those of you who may not be familiar with Ovid Therapeutics, just a little intro to ourselves. So we are a small neurotherapeutics company with a passion for and a focus on diseases with significant unmet need. And doing that, we believe that by leveraging our academic partnerships and partnerships with patient advocacy groups, we can help to accelerate therapeutics and drug discovery for these rare disorders. So we have, Ovid Therapeutics has basically two arms. We have an epilepsy franchise where we are developing programs for specifically for targeting seizure disorders. And we also have a genetic medicine arm, and that's a little bit about what I'm going to tell you today. Next slide. Thank you. So we at Ovid believe that by working together with the patient communities from the very beginning of the drug development process, we are going to accelerate these potentially life-changing therapies. So we're lucky to work with individuals like Luke Rosen and Sophia Cacciatore, who are part of our group and part of your group, to really engage with the patient community from the very beginning of our process. Our goal is to develop medicines with a clinically relevant endpoint. And so in order to do that, we want to be really involved in the natural history studies that Dr. Chung mentioned earlier. We want to be able to be ready to move these therapeutics forward with meaningful measures at the end. Next slide. So we do have this excellent collaboration with Wendy Chung, who needs no introduction to this group. She has a fantastic team at Columbia University who have done extensive work into developing induced pluripotent stem cell models and animal models for KIF1A associated disorder. They have the ability to use these tools for screening compounds and for screening genetic therapies. And together, by working with Wendy, who also is involved in the natural history studies, we believe that we're really setting ourselves up to be able to translate and accelerate our discoveries into human trials. Next slide, please. So just to give you sort of an overview of what this drug discovery process looks like and how we're working together in this synergistic development plan with Columbia, I'll just take you through kind of a high-level overview of the process. So we're currently in the stage of discovery, and this is working very closely with Wendy Chung and her excellent research team. A few of them are in the audience today. Also, I just want to call them out because the work that's ongoing is really, it really is on their shoulders, and they're doing a fantastic job. Charles Leduc, Mike Zucchero, and this is work that's also started with Leah Boyle. We are proud to be part of this collaboration. So Wendy's team is using, as I said, the induced pluripotent stem cell models, which are able to be turned into neurons and astrocytes, cells representative of the central nervous system. And they use these model systems to be able to test various RNA targeting modalities that I'll talk to you a little bit about today. The work that's ongoing there is looking at identifying potential drug candidates, being able to investigate ways to deliver these drug candidates and validate them in these phenotypic assays that will allow us to say that the drugs that we're developing are fixing a functional assay in these human cell models of CANT. And ongoing at the same time are those critical natural history studies that we mentioned earlier. Where Ovid's next step comes in is once we've gotten to what we call the lead ID. So all of the work that's ongoing to discover the molecules that would be potential drug candidates, when we identify one of these candidates that we have really, really strong in vitro data on, we nominate that as our lead candidate. And that's taken into sort of the drug development process. So there's the discovery and then there's the development. In the development process, these lead candidates are taken into translational in vivo models. So whether those are mouse models, whether those are rodent models, non-human primates, there are certain studies that need to happen that will lead to understanding of the distribution of these drugs throughout the body, the safety, most importantly, of these drug candidates. And then another important step is manufacturing and being able to scale up the drug candidate for actual moving towards delivery in human. And so these steps are a critical part of the process. They also allow us to perform certain IND enabling studies. The IND process is basically this massive document of all of the information that's gone into the discovery and development of this drug. That is filed with the FDA, and that will allow us, if approved, to move the drugs safely into first-in-human trials. So it's really hard, as you heard from some of the previous speakers, to put a timeline on this process. Our best estimate at this point would be to tell you that once we've gotten to that point of lead ID, we've finished the discovery part of the process. It's about 18 to 24 months of work that has to go into the IND enabling studies, the safety studies, before anything can be moved into a clinical trial. Next slide, please. So as you've all heard and may be aware, DNA sequencing is unlocking so much potential in developing therapies for rare genetic conditions. So these advances have accelerated the identification of monogenic disorders, so conditions where we know exactly the root cause of that disorder, so the particular gene mutation that causes the symptomology of a disease. With that information, there's a lot of power in being able to develop a genetic therapy. So Ovid's approach is really to perform an RNA-targeted approach. So you may have heard of gene therapy in the context of replacing a healthy copy of a gene, for example, in a condition where a mutation causes a missing protein in the cells. So Ovid is actually not taking a gene replacement approach, but rather an RNA-targeted approach. And so what how this works would basically be, if you think about the gene is the instructions for a protein, which is the functional workhorse in the cell. The intermediate there is what's called RNA, and RNA is sort of like the messenger. RNA is transient, and so it can be tackled in a few different. RNAi or RNA interference is traditionally accomplished using what's called an siRNA, which is a small interfering RNA, or a vectorized, meaning carried in by an inactive virus, shRNA or short hairpin RNA. Another approach that you've probably all been hearing about is antisense oligonucleotides, or ASOs. All of these approaches basically aim to degrade a copy of the RNA in the cell, and that ultimately will lead to a loss of that defective protein in the cell. So we're interested in developing RNA approaches for gain-of-function mutations in CAND. Next slide, please. Okay. So the reason that we can do this, as you heard from Dr. Chung earlier, is that there are some variants, some mutations in KIF1A cause a dominant negative gain of function. So just to take you back to what the KIF1A, I actually just recently heard a really great description from Dr. Mike Zucchero about how we can envision what KIF1A does. So if you envision that half of the KIF1A dimerized functional protein comes from one individual contributing one hand and one foot, the other part of the dimer is another hand and another foot. And together those things comprise the body of KIF1A protein that's able to carry the cargo, the synaptic vesicle cargo, along the microtubules to the synapses and deliver it for a function of the neuron. So what happens in the case of a dominant negative is that half of that body has a defect, is inhibited, and is not able to participate in the transport of that cargo. So what Ovid's approach is to basically expression of that mutant half of the protein. So if we can get rid of the arm, the hand and the foot for the mutation, you can actually allow then only wild type or healthy KIF1A to dimerize and recover function and goes. Next slide, please. Okay, so as I said, Wendy Chung's team is working really hard on characterizing iPSCs and neurons derived from them from patients with various KIF1A mutations. And so they are developing phenotypic screens. You've heard of some of these from Dr. Wayne Poon. They include, the neurite outgrowth phenotypes, neuronal differentiation phenotypes, looking at what's called electrophysiology, so the actual electrical firing of these neurons and how they communicate. Importantly, for KIF1A, we can look at kinesin aggregation and actual transport of synaptic vesicles. And what you're seeing here on the slide is some data from Dr. Leah Boyle, comparing neurons derived from individuals with 2 of the KIF1A variants to a wild type neuron, characterizing some of the deficits that these neurons may have. Next slide. So I wanted to take you through sort of the, using some excellent slides that were put together by Leah Boyle in the Chung lab, the genetics that are underlying the dominant negative variants and how we think our RNA targeting approach can actually benefit. So to start, in the case of an individual with 2 healthy alleles, so what that means is 2 healthy copies of the KIF1A gene. What happens is that the RNAs that are made as copies of those genes are turned into protein, and what you have then are functional dimers with normal healthy KIF1A protein. So that's kind of depicted in these little molecule schematics where the white are healthy KIF1A protein. What happens in the case of a nonsense mutation or a loss of function mutation is that one of the alleles does not make any RNA, does not make any protein. So in these individuals, they would have about half of the normal amount of KIF1A protein, and that's represented here by 2 dimerized KIF1A molecules. In the case of the missense mutations, these are mutations that would cause potentially gain of function variants. What happens is that one of the alleles, the missense variant, and that causes a dysfunctional portion of the KIF1A dimer. And so in these individuals, about 25% of the protein would be dimers that are normal, so healthy, and those would be functional KIF1A proteins. In some of the cases, you would have 1 mutant KIF1A dimer, part of the dimer, and 1 healthy, and this is expected to contribute an inhibited function, a disrupted function of the KIF1A motor. And in some cases, you would have dimerization of 2 mutant copies, and we would expect that to be even more of a deficit to the gene. Next slide, please. And so the allele-specific RNA-targeted approach that Ovid is taking with Columbia University is to try to interfere with expression, the message coming from the missense allele. So again, now we're looking at more of these hypothetical proteins here. In the ideal situation at the top of the slide, you would have healthy dimers of KIF1A and totally functional normal protein. In the case of heterozygous missense variants, again, you would have 50% of the KIF1A monomers would have a deficit, and that would interrupt some function when they dimerize with wild type healthy monomers. If we're able to reduce one or the other of these variants, what we would expect in the called out blacked out box here, if we can specifically reduce expression or RNA and then protein coming from the missense variant, we can increase the number of dimers that are coming from healthy monomers of KIF1A and potentially improve the overall KIF1A function. And so working together with Wendy's team, I don't have the data to show you today, but we have identified oligos that are specifically able to reduce RNA coming from the KIF1A variant alleles and not affecting or very much less affecting the wild type alleles. And we're continuing to investigate the biology of delivering these RNA targeted approaches. and as well as moving these into the phenotypic assays to be able to show functional rescue when we reduce the specific KIF1A variant. Next slide, please. So as you heard Dr. Chung introduce earlier this morning, the KIF1A allelic spectrum, so that the number of variants that occur in KIF1A is vast. And so in order to develop an oligo approach, so when I say oligo, I mean either an siRNA, an shRNA, or an ASO, in order to develop one of those drug products for each and every variant that causes CAND in individuals is a very immense undertaking. And so we've been thinking very hard about how can we approach many different variants with very much fewer drugs. And so this is a thought process coming mainly from Wendy's team and from Charles Leduc with this awesome schematic here, where we're taking advantage of other variants that occur distributed throughout the genome in healthy individuals. So these are what are called single nucleotide polymorphisms, and they occur throughout the genome, as I said, and they do not cause any known phenotype. So these are synonymous variants. What they are is places in our genome where different individuals can have a single different sequence, single different base pair, an A, T, C, or G in the DNA. And so what we want to do is basically look at correlating where these SNPs are with being on the same RNA strand as a KIF1A CAN-causing variant. And so if we can identify a subset of these synonymous SNPs that we can use as a handle, if we can develop an oligo drug against that handle, and that same handle would be shared by individuals with 3 or 4 different KIF1A variants, we can develop fewer drugs and target a larger population. And this is work that's ongoing in our collaboration that we're very excited about because we believe it'll help us to be able to develop therapeutics for a larger group of patients. Next slide, please. And so I just wanted to also introduce to you all an additional model system that we're working with. So Wendy's team has these excellent iPSC models and are, in addition, are characterizing mouse models. We're also support this process by developing a worm model. So worms are a lot easier to grow in the lab, and they, interestingly, the fate of every single cell in a worm embryo has been characterized and understood what the ultimate goal and function of that cell is in the functioning worm. And so what we want to do is to be able to analyze KIF1A variants in the context of a worm so that we can use them to actually study functional rescue and the function of these variants in a really high throughput way. And so some of this work, as kind of shown on these slides here from Lam et al. In 2021, and Onizawa also in 2021, this work is kind of instrumental in showing that you can model the KIF1A variants in the worm. And we're also building a model at Ovid in the worm so that we can screen our oligo compounds that are coming out of the Columbia collaboration to see whether we can fix these deficits that are coming out of the worm models. Next slide, please. So as we're preparing all of this work, this preclinical work that's ongoing with Wendy's team and Ovid's support there, we're also paying attention to what is happening in the landscape of genetic therapies. We want to be prepared to be able to move forward any oligos that we are identifying as quickly and as safely as we can. In an effort to be ready to go, we are paying attention to the learnings that are happening in the field of genetic therapies. So recently, Ovid joined what's called the Bespoke Gene Therapies Consortium. So the BGTC is largely a partnership between the NIH, the FDA, many different pharmaceutical companies and nonprofit organizations. And their ultimate goal, the ultimate goal of this consortium, is really to create resources that will allow the streamlining of genetic therapies. So understanding the various components that go into developing a gene therapy, including the vectors that are used for delivering them, the process of manufacturing these gene therapies, the necessary preclinical testing and animal testing, toxicity testing that has to go into making sure that the process is streamlined for bringing these therapies from basic discovery into the clinic. And so we hope to learn a lot from being part of this consortium and being ready to move our therapies forward. We're also paying attention to the recent guidelines that are coming from the FDA in the development of oligonucleotide therapies, whether this be an siRNA or an ASO. The FDA, it's exciting that they've been releasing recent guidances for helping drug companies and clinical trials really understand the process of what's needed to determine safety, pharmacology, and being ready to really move these therapies forward in the best way possible. Next slide, please. So in summary, I've told you today just a little bit about how we're working with the Columbia University team together with Ovid to create screening models, functional models for being able to study KIF1A. We're using these models to screen for allele-specific or mutation-specific RNA interference sequences. And these sequences are being screened for validation in these phenotypic models that are under development. We are leveraging the genomic sequencing data to identify those SNP handles that I mentioned, where we're hoping to be able to target more KIF1A variants in a more efficient way. And we hope to be able to move these development process not only for KIF1A, but to support, as Ovid as a whole, to support many different monogenic disorders and kinesiopathies. So I want to just take a second to acknowledge the team. So I want to acknowledge Patrick Sarmiere, who is here and I think will join me for the Q&A. He is VP of Preclinical Research at Ovid. Tae-Hoon Kim, who is our neuroscientist expertise within Ovid. Luke Rosen and Sophia Cacciatore, who are in our patient engagement, community engagement at Ovid. Want to thank Wendy and her team, in particular Leah Boyle, Charles Leduc, Jingquan Hu, and Mike Zucchero, and also consultants and collaborators that we work with, Arna Gendrich and Jim McSwiggan. And with that, I thank you, and I'll take some questions.